was noticed. So it was very fascinating to note that out of 30 inbred line, 8 inbred line named M2, 4, 5, 6, 7, 9, 17, and 22 has given the lower fungal colonization in the rating scale of 1 to 5. And this almost same pattern was noticed when these inbred lines were treated with AF3 isolate, which has been collected from the Assam region. So it gave us a ray of hope like among these 13 inbred lines, we have some lines which really have the good genetic background. So further, we taken up the ELISA testing for aflatoxin B1 molecule in our laboratory, and it was again interesting to note that out of this 30 inbred lines, M1 to M30, the same line which has given the lower fungal colonization in kernel screening has given the lower OD value at 405 nm. It means it having the capability to resist the aflatoxin B1 buildup. So after this ELISA testing, the from kernels, the DNA was isolated, and so far there is no report for this trypsin inhibitor gene in the maize, although reports are there in other hosts sorghum and panicum. So based on this idea, we have designed one primer for the trypsin inhibitor gene, and among this 30 inbred line, again these 8 lines, that is M2, 4, 5, 6, 7, 9, 17, and 22, gave amplification at 396 base pair for the trypsin inhibitor gene presence, and the evolutionary study clearly shows that these genes fall in a different clade or cluster. It is entirely different from the same trypsin inhibitor gene present in the sorghum host as well as panicum host. So this is the Western blotting and STS test was performed after this PCR assay. So protein was isolated from this promising 8 inbred lines in the laboratory. The protein profiling was tested in the STS page. Later on, specific trypsin inhibitor antisera was used to carry out Western blotting, and the presence of 14 kilodiameter trypsin inhibitor protein was noticed in these promising M2, 4, 5, 6, 7, 9, 17, and 22 inbred lines. So this has also come as one of the good report for the particularly from the maize host. So for trypsin enzyme assay was later carried out. These lines have the better performance for the trypsin inhibitor enzyme. So after this in vitro studies, what we did, we performed this inoculation artificially with two different inoculation methods, world inoculation and the injection of spores inoculation. In world inoculation, 49 days old plant were used where 1 gram of inoculum was placed, whereas in injection was done with 9 into 10 to the power 7 CFU per ml of spore suspension at the primary air stage. So it was very surprising to note that although these 8 lines M2, 4, 5, 6, 7, 9, 17 and 22 has given the better promising performance or resistance performance in the laboratory testing, whereas in the artificial inoculation among these 8 lines there were 3 lines M2, 4 and 5 have developed the symptom in the maize. This is very conspicuous here, noticed for these 3 lines, and only 5 lines we end up which has given the better performance in laboratory testing as well as field testing. So I would like to conclude that among these 20 fungal isolate AF1 to AF20, there are two isolates which has been collected, one from South India, this AF1, and another from Assam. They are highly toxigenic and they amplified for all the aflatoxin producing genes of their pathway, structural gene as well as regulatory gene. And these two aflatoxigenic isolates were used for the study, and apart of this 20 isolates, the rest 8 isolates were ataxogenic, which is used as a biocontrol agent. And among this inbred line, there were 8 lines M2, 4, 5, 6, 7, 9, 17, and 22 has the presence of trypsin inhibitor gene, which is reported as one of the resistance factor. In other hosts, in maize also, it has shown the best performance. And apart from this whole study, in vitro as well as in vivo study, we end up with the 5 lines M6, 7, 9, 17, and 22, which has the very good genetic background for aflatoxin B1 development, as well as these lines have the good resistance response, and these materials are under the process of registration in ICR NPP here New Delhi. And apart from this work, there is Another many more work can be done. The future prospect include the mechanism of biocontrol activity of the fungus, as well as what are the underneath mechanism involved for aflatoxin B1 development. And there is a need to develop a rapid, sensitive, efficient, and very eco-friendly tools for the detection of these molecules, which is a major food contaminants in the food stuff. And I would like to acknowledge Director IRA and BPGR New Delhi, Head Professors, Division of Plant Quality, Scientist Division of Plant Pathology, IRA New Delhi, my senior, juniors, lab mates, SRF, JRF, and other all supporting staff who helped me to carry out this work. At the end, I would like to tell a little awareness in farming community will give healthy and sufficient food to all in stomach. Thank you.
Sir,
Thank you,
she has
Payal.
taken 13 minutes for completion.
Thank you.
Thank you, sir.
Now I will call upon Shreya M. Joshi.
Yes, sir. Sir,
So
shall I put
Shreya,
my PPT?
you share your screen.
Yes, I'll share my screen. Is it visible now, sir?
It's
Yeah, you
visible,
share
but
the
in the
slides.
window. Yeah, you extend it. Yeah, it's
Yeah.
visible.
Can you see the slides, sir?
Yes, yes.
So shall
So
I start?
you can start your session.
Yeah. Good afternoon, everyone. I am Shreya Joshi from Karnataka University, Dharwad, and it gives me an immense pleasure to present my PhD work on microfabricated selenium nanoparticles mediated proteomic and transcriptomic alterations as a defense response against late blight of tomato as a part of the prestigious P.R. Verma Award for the PhD students. As we all know, tomato is an economically important crop and the global tomato production is approximated up to 130 million tons. India, being the second largest producer of the crop, produces up to 19.4 million tons of tomatoes, which is cultivated in an area of 7,86,000 hectares. The major tomato producing states in India are Andhra Pradesh, Karnataka, and Madhya Pradesh. It is found to be rich in different nutrients such as vitamins A, B, and C, and a very important antioxidant molecule that is lycopene, which is involved in providing protection against cancer. However, the cultivation of tomato is being challenged by different biotic stresses such as Alternaria stem canker, anthracnose, early blight, late blight, etc. But in the year of 2015, severe outbreak of late blight caused by Phytophthora infestans was reported in the southwestern parts of Karnataka that caused up to 100% yield losses. This late blight of tomato, as given by my fellow participant, Mr. Mahendra, has already briefed upon the symptoms of the late blight. So I'll just skip this one. Coming to the challenges in controlling late blight of tomato, conventionally the fungicides being employed are dimethamorph mancozeb, phosphatile aluminum, azoxystrobin, etc. These control measures, being chemical in nature, pose a threat to the environment as well as to the human health. Moreover, the sexual reproduction of the pathogen has also allowed the evolution of newer lineages that are resistant to the conventional fungicides used. Thus, it forms a very imperative need to develop eco-friendly alternatives that can effectively combat Phytophthora infestans. Solution: it can be provided by the upcoming technology like nanotechnology, wherein the nanoparticles possess a minute size which renders them a large surface area to volume ratio. The nanoparticles also possess a better contact with microorganisms with enhanced antimicrobial activity. Also, because of the greater solubility and more stable dispersal of the components, there is a chance of decreased persistence in soil, thereby reducing the chances of environmental pollution. And in this context, selenium has also been found to inhibit the spore germination of fungal pathogens by accumulation of reactive oxygen species and induction of resistance against fungal phytopathogens. Another sector of disease management for plant diseases is through PGPFs, that is the plant growth promoting fungi, which have a high antagonistic activity through the production of antimicrobial compounds, hydrolytic enzymes, and they are also found to induce systemic resistance in host plants against different biotic stresses. This study was targeted to combine the effects of the nanoparticles and the PGPFs in the form of micronanoparticles, which were employed in the present study. Scope of investigation: as discussed, tomato is an important economic crop, and the late blight of disease has posed serious challenges for the disease management practices. There have been many reports on in vitro and in vivo activities of synthesized nanoparticles in triggering the antioxidant enzymes, defense enzymes, and pathogenesis-related proteins upon priming. But there are very few reports regarding differentially expressed proteins mediated by trichoderma-derived selenium nanoparticles. Thus, this work will allow me to bridge this gap and understand the modifications of the host mediated by the biosynthesized nanoparticles to induce resistance and yield high-yielding tomatoes. This is the summarized workflow that you can see here. Initially, the selenium nanoparticles were synthesized by subjecting different fractions of the trichoderma, that is Trichoderma atroviridae, with 25 millimolar concentration of sodium selenide, and the reaction mixture was observed initially for the color change. The synthesized nanoparticles were then undertaken for their in vitro antifungal activity against different fungal phytopathogens. The bioactive selenium nanoparticles were then characterized for different physicochemical properties based on different techniques such as Fourier transform infrared spectroscopy, scanning electron microscopy, energy dispersive X-ray spectroscopy, high resolution transmission electron microscopy, and powder X-ray diffraction studies. These bioactive anti-characterized selenium nanoparticles were then used as nanopriming agents of tomato seeds, and induction of systemic resistance was evaluated under greenhouse conditions to evaluate the growth and development parameters of tomato, and also to evaluate the plant protection, the percentage of plant protection against late blight of tomato. Further, the study was extended to quantify different biochemical defense markers such as phenylalanine lyase, beta-1,3 glucanase, superoxide dismutase, and lipoxygenase, which were further validated by transcriptomic analysis. Lastly, the study was aimed to profile the differentially expressed proteins in selenium nanoparticle treated plants against Phytophthora infestans by using SDS-based two-dimensional electrophoresis and mass spectrometry. Coming to the microsynthesis of selenium nanoparticles, after a period of 24-hour incubation, which consisted of different fractions of the Trichoderma atroviridae fungus, a color change from colorless to dark brick orange after 24 hours was observed. These formed selenium nanoparticles were then evaluated for antifungal activity against Pyracularia grigia, and it can be observed that at 200 ppm concentration, a maximum inhibition of the fungal growth was observed that was recorded up to 52.33%. Further, the in vitro leaflet assay was carried out wherein the chili leaves and the tomato leaves were pre-treated with different concentration of selenium nanoparticles, and further the leaves were treated with Coledotrichum capsicum on the chili leaves and Alternaria solani on the tomato leaves. As compared to the control, it can be seen that different concentration of selenium nanoparticles could inhibit the growth of the pathogen on the surface of leaves as compared to the control, and at 100 ppm concentration, the total spread of the inhibition was inhibited. Further, we evaluated the direct inhibitory action of selenium nanoparticles against the zoospores of Phytophthora infestans, wherein the spores were subjected to 25 millimolar to 200 ppm of selenium nanoparticles, and the reaction mixture was incubated for 1 hour in dark. It can be seen that as against the control, in the treated spores which received 100 ppm of selenium nanoparticles, extensive aggregation of spores were observed, which were further validated by atomic force microscopy and scanning electron microscopic techniques. These bioactive nanoparticles were then characterized for different physicochemical characteristics, such as energy spectroscopy for the formation of selenium nanoparticles, the DLS to measure the Z average size and zeta potential, FTIR to identify the functional groups associated with selenium nanoparticles, X-ray diffraction studies to establish the crystalline nature of nanoparticles, and electron microscopic studies to study the surface morphology, elemental composition, and size of the nanoparticles. From here, it can be seen that the DLS revealed the Z average size of the nanoparticles ranging from 93.2 nanometer to 98.5 nanometer, and a very high zeta potential of minus 49.3 millivolts was observed. This indicates a high stability of the nanoparticles owing to the electrostatic repulsions between the selenium nanoparticles. And from the UV spectroscopy, it is a characteristic peak at 260 nanometer was observed, thus confirming the formation of selenium nanoparticles. So here is the FTIR and the XRD spectrum that you can see here. The FTIR spectrum showed hydroxyl groups and amine groups involved in the reduction process of sodium selenite that is present in the +4 oxidation state to the elemental selenium in the form of selenium nanoparticles. The X-ray diffraction pattern of the microgenic selenium nanoparticles is presented here, where different peaks were observed that corresponded to different sets of lattice plane, establishing the crystalline nature of the selenium nanoparticles. The electron microscopic images you all can see here. The shape of the selenium nanoparticles were found to be spherical, with selenium as the major element present in the nanoparticles. And by the HR-TEM studies, it was evident that the sizes ranged from 60.48 nanometer to 123.19 nanometer. These bioactive characterized nanoparticles were then used as nanopriming agents, wherein 4 different treatments were considered to compare selenium nanoparticles efficiency. The first set of the seeds were treated with sterile distilled water. The second set of seeds were treated with the pathogen, that is the Phytophthora infestans. The third set received only selenium nanoparticle treatment with 100 ppm concentration. And the last set received both selenium nanoparticle and the Phytophthora infestans that served as the tripartite interaction in the present study. These treatments were then evaluated for plant growth and development based on these factors, and also reduction in disease incidence was recorded. As you all can see here, as against the control, the selenium nanoparticle treated tomato seed showed 91% of early germination, a higher seedling vigor, almost 100 centimeter higher plant height was observed in treated seeds. The treated seeds flowered almost 10 days earlier to the control ones, and the fruit weight was also found to be higher. The priming efficacy of selenium nanoparticles to tomato seeds and the duration required for induction of protection against late blight disease under greenhouse conditions was evaluated. It was seen that at the second day after the root tip inoculation of the pathogen, 72.9% of disease protection was observed. These greenhouse data were then taken into the laboratory conditions, where we assayed different antioxidant and defense enzymes such as lipoxygenase, phenylalanine lyase, beta-1,3 glucanase, and superoxide dismutase. As it is evident from the graph, at 24-hour and 48-hour post-inoculation, all the enzymes were found to be upregulated, and this activity was quite stable even after the decrease of the time, thus indicating that the selenium nanoparticles induced the activity of these enzymes when infected with the late blight pathogen. So these enzyme activities were further validated by transcriptomic analysis by analyzing the expression of mRNA upregulation by using these different primers. And here we have presented in the form of graph, where it is quite evident that as similar to the enzyme activities, the upregulation of the mRNA expression was parallel. At 24 and 48 hours, the highest upregulation was seen, which was also stable at later time points. Lastly, the last aim of the work was to profile the total proteins from the 4 different treatments to reveal the differentially expressed proteins. The preliminary STS page revealed distinct unique bands that were not present in any other treatments. These unique bands were present in the treatment which received both the nanoparticle, that is the N, and the pathogen. Further, the differentially expressed proteins were then profiled by 2-dimensional electrophoresis, wherein the treatment which received selenium nanoparticle and Phytophthora infestans inoculation showed accelerated upregulation of many distinct proteins, some of which I could not mark here, indicating the induction of resistance by the nanoparticles. Lastly, these differentially expressed proteins were then identified by LC-ESI-MS-MS, wherein 5 different proteins were observed, that is mainly fructose bisphosphate aldolase, chromatin-like protein, superoxide dismutase, bibulase bisphosphate, and glycoproteins. These are found to be involved in different important functions such as defense, response, photosynthesis, antifungal properties, etc. So coming to the discussion, this present study highlighted the broad spectrum antagonistic activity of Trichoderma atroviridae selenium nanoparticles. For the very first time, we have demonstrated the aggregation of Phytophthora infestant zoospores when treated with 100 ppm of selenium nanoparticles, which was visualized by different microscopic techniques. The selenium nanoparticles prime plants showed a positive effect on growth and development of tomato plants. The selenium nanoparticle induced plants also exhibited a significant resistance of up to 72.9% against late blight disease. The induced resistance was positively correlated with corresponding alterations at biochemical levels at different time points. In addition, we have also noticed the upregulation of defense related genes in prime plants in comparison with the respective controls. And lastly, 5 differentially expressed functional proteins were detected by LC-SIMSMS that play a vital role in the establishment of plant defense. Overall, the present study represents an orchestrated correlation between the resistance and defense responses incited by the selenium nanoparticles against tomato late blight disease, which can be used as a nanobiostimulant in protecting tomato plants. So coming to the conclusion, it can be stated that the microfabricated selenium nanoparticles offer a novel tool for crop protection strategies. Thank you so much, everyone.
Sir, she has taken exactly 15 minutes.
Oh. So, thank you, Shreya.
Thank you, sir.
Now I will call upon Deshraj Sri Bharti. Deshraj, can you hear me? Deshraj Bharti.
Deshraj Bharati. I think he is not there, sir. You can call the next speaker.
Okay, next, Archana TS.
Yes, sir.
So, are you ready to present? Share your screen.
Yes,
Share your screen then.
yes, sir. Sir, is it visible now,
So,
sir?
yeah, it's visible.
Okay.
Thank you, and your time starts now.
A very good afternoon to one and all. I am Archana TS from the Department of Plant Pathology, here to present my doctoral work carried out on molecular characterization of pathogen in the complex and interaction causing rhizome rot in ginger. The present work has been carried out with the guardianship of Dr. Aki Mehta. So, as we all know, ginger is one of the most important spice crops cultivated in India. Even though several foliar diseases exist in this particular crop, rhizome rhotis ginger is very important because of the severe yield loss. And the complex nature of the pathogen make the management strategies very difficult. Till now, no effective management strategies are available. Considering all this, we have set the objectives like molecular characterization of the pathogens which is involved in the complex, interaction studies of the pathogen, evaluation of different elicitors for the receptor induction, and screening of the germ plasma for resistance, and finally coming up with a integrated disease management module. The different isolates we have collected from the major ginger growing areas of Kerala and Karnataka, and this reveals the pathogen complexity of the different pathogens which is involved in the disease, like Ralstonia solanacearum, Fusarium species, Pythium aphanidermatum, Sclerotium rolfici, and Meladogina species. These are the microscopic observation of different Pythium isolates which I have collected from the different parts, and these are the Fusarium isolates and the microscopic view. And this is the Sclerotium rolfici, and it is a media to distinguish the virulent and virulent colonies, and an electron microscopic study to understand the structure and the shape of the bacteria and the perennial pattern of the corresponding Meladogina. So even though it is a complex disease, individual pathogens are capable for producing typical symptoms. Where Pythium aphanidermatum cause like initial symptoms like yellowing followed by the complete rotting of the rhizomes. Whereas in case of Fusarium, it causes wet yellows or wet rot followed by the initial yellowing, foliar yellowing, complete drying, and finally discoloration of the vascular bundles. Whereas in case of Sclerotium rolfici, it also starts with foliar yellowing, drying, and complete rotting of the further rhizomes. Whereas in case of bacteria, yellowing and drooping followed by the rolling up, and finally the infected rhizomes completely rot and produce foul smell. In case of nematode, the initial symptoms in the field level appears as chlorotic yellowing, and finally appearance or formation of galls. The pathogenesis of all the individual pathogens were proved individually, and the same symptoms which I have mentioned before were reproduced, and the pathogens were re-isolated. Further, the molecular characterization of the pathogens were carried out by using species-specific primers and revealed that the pathogen mainly belonged to Pythium aphanidermatum, Fusarium solani, Fusarium oxysporum, Ralstonia solanacearum, and Sclerotium rolfisi. Whereas in case of Melodogyna, the DNA were isolated and sequenced, which revealed that it showed high similarity with Melodogyna hapla as well as Melodogyna enterlopi. From here, it is very clear that there is an association of different pathogens which is involved in the disease complex. Further, to understand is there any interaction between the pathogens which cause the disease complex, an interaction study were carried out by individual treatments as well as simultaneous inoculation of all the pathogens and combination of different pathogens. And the results revealed that among the individual pathogens, Ralstonia folaneacearum found more potential for causing the disease. And the simultaneous inoculation of all the pathogens showed higher severity of disease incidence as well as rhizome rot. Among the sequential inoculation of different pathogens, Melodogyna followed by Ralstonia showed higher percent disease incidence as well as rhizome rot incidence. The same result we have depicted when we have inoculated the plants after 105 days after planting, and these are the pictorial overall representation of the plants which have treated with the different combination of pathogens. Here represents the intensity of Ralstonia solanacearum which causing the diseases, and here it represents the simultaneous inoculation of all the pathogens. Among the different sequential inoculation of different pathogens, Melodogyna followed by Ralstonia caused a higher percent disease incidence as well as rhizome rot incidence. The percentage of rhizome rot incidence also we have evaluated under the conditions, and the same result which have given before we have interpreted, and among the individual pathogens, Ralstonia found more potential for causing the pathogens, and as a combination of different all the pathogens inoculation and the sequential inoculation, Melodogyna followed by Ralstonia. The same result have interpreted when we are treated the plants after 135 days of planting, and Melodogyna followed by Ralstonia found more potential for causing the disease incidence. So this is the overall representation of percent disease incidence after different time period of like 75, 105, and 135. Some compared to the all the days, 135 found more potential for causing the disease, simply indicating that it is a very vulnerable stage for the pathogen to attack and cause very high disease incidence as well as rhizome rot. Even though the individual pathogens are capable for causing the disease, the simultaneous inoculation as well as the predisposition of the nematode, which highly increases the disease severity as well as rhizome rot. The same results we have depicted in case of rhizome rot, and in case of ginger, there is no such resistant varieties is available. And to evaluate the external application of elicitors can induce any type of different response in ginger, an evaluation study has been carried out by using different elicitors. Preliminary study has been carried out by the in vitro evaluation of different elicitors against all the pathogens which causing rhizome rot, and none of the elicitors showed any kind of inhibition against the pathogen complex. However, biocontrol agents like Pseudomonas fluorescens and Trichoderma hertzianum found potential for inhibiting the pathogen's growth. And further, these are the different pictorial representation of evaluation of elicitors against Fusarium, against Pythium, against Sclerotium rolfisi, and the Ralstonia solanacearum. The experiment has also taken under the greenhouse condition in order to understand the efficacy of elicitors under the pod culture, and the results revealed that methyl jasmonate found very potential due to reducing the incidence of the disease as well as rhizome rot incidence, followed by salicylic acid. The same results we have interpreted or got when we have treated the plants in a curative way. To understand the nature of the different response genes after the elicitor treatment, an expression study were carried out by treating the plants with elicitors, followed by the sequential inoculation with pathogen, and subsequently taking out the samples at different period of time, followed by the RNA extraction, cDNA synthesis, and qPCR assay. So these are the different treatments we have imposed under the pod condition, and these are the 10 different genes which we have targeted, like majority of the genes which have involved in case of different response pathways, isopropanol pathways, biotic and abiotic stresses, stress tolerance, as well as antifungal activity. So these are some of the amplification part of the respective different response genes, and here it represents a single MEL curve which we have which we received from the different response genes. And The external application of different elicitors were carried out like a prophylactic and curative way, and 10 different genes which are involved in the defense pathway were evaluated. And here, in case of external application of elicitors as a prophylactic way, initially all the genes were expressed in the early hours itself, and in case it has maintained the expression even after the 10th day after pathogen inoculation. However, application of treatment 4, that is methyl jasmonate, result higher expression of the genes during the 8 hours after pathogen inoculation and maintained even after the 10th day. However, in case of curative, less expression of the genes were reported. Where in case of HMGR, application of eugenol resulted the higher expression of the genes from the early hours itself and peaked in the 8 hours. As compared to the prophylactic, curative samples showed less expression of the genes. Whereas in case of HMGS, the early expression of all the application of elicitors resulted in an early expression of all the genes, whereas in curative also the expression of genes were more. As compared to other different response genes, application of elicitor resulted higher expression of MLO genes which is involved in the different response, both in case of prophylactic as well as in case of curative. Whereas in case of Ricky transcription factor, application of Trichoderma hertzianum resulted in the early expression of the different response genes, and even it is peaked after 24 hours, and the expression has been maintained even after 24 hours. Whereas in case of curative, as compared to the prophylactic, all the genes were expressed in higher amount. The same result has been expressed in case of HSP, where the external application of Pseudomonas fluorescens from the initial time period itself and up to the 10th day of pathogen inoculation, higher expression of the genes were maintained. Whereas in case of curative, application of Trichoderma hertzianum resulted the early expression of the genes and even maintained after 10 day of pathogen inoculation. Whereas in case of callus in place, all the application of elicitors resulted the higher expression of the genes both in case of prophylactic as well as in case of curative. Same result we have depicted in case of glucanase. The application of all the elicitors resulted higher expression of the genes which is involved in glucanase synthase, both in case of prophylactic as well as curative. Where we come into the PR protein 5, application of eugenol resulted in the early expression and peaked up to 8 hours, and further down expression of the gene has been reported. Whereas in case of curative, all the elicitors comparatively or significantly showed higher expression of the gene. So this is the heat map which shows the expression of different elicitors under prophylactic as well as curative conditions, where the green color which represents a higher expression of the genes, and the red color which indicates lower expression of the genes, and intermediate color which represents a moderate expression of the genes. We have also evaluated 14 different ginger varieties under different conditions in order to understand the nature of resistance. And among the 14 different varieties we have evaluated, Neelamaren found partially tolerant, partially resistant against the disease. However, when the same variety, when you have evaluated in the in vitro or in the pod condition, it is found partially susceptible. We have also used different effective molecules in order to come up with an effective management, integrated disease management module. For that, first we have used different nanoparticles, that is copper and silver nanoparticles, in order to evaluate the efficacy under the conditions. And among the different nanoparticles, copper nanoparticles, especially 100 ppm, showed significant reduction in the pathogen drop. The same result we have obtained when we have used silver nanoparticles against all the pathogens which is involved in the complex. And when we are used copper nanoparticles against the pathogen, discoloration of the mycelial mat were observed. In order to understand the mechanism behind that, a detailed electron microscopic study were carried out, and these are the treatments with nanoparticles and without nanoparticles. So the treatments after nanoparticles show significant structural disturbance as well as destruction of the mycelial mat, and the treated nanoparticles fungus were failed to produce spores as well as oospores or microponeria and macroponeria. So this has happened in case of fusarium. So this is the picture with nanoparticles and without nanoparticles and with nanoparticles. And after the nanoparticles treatment, destruction as well as structural integrity of the mycelial mat were highly hampered. Whereas in case of bacteria, after the nanoparticle treatment, the cell destruction as well as complete lysis of the bacteria were observed. We have also evaluated the nanoparticles under the pod conditions, and 100 ppm concentration of the nanoparticles found effective in reducing the disease incidence as well as rhizome rot incidence. The same result we have got when we are used silver nanoparticles for the management of the disease. We have also evaluated different antibiotics as well as combination of antibiotics and agrochemicals. So among all the antibiotics evaluated, streptocycline found effective, and we are also evaluated different non-systemic chemicals, systemic chemicals, as well as combi products against pythium, fusarium, and sclerotium. So these are the in vitro evaluation of different non-systemic, systemic, as well as combi products against pythium. So these are the evaluation of different systemic, non-systemic, as well as combi products against fusarium. And these are the systemic, non-systemic, and combi products against sclerotium talcicum. And those the treatments which have found better or superior results under the lab condition, we have evaluated under the field condition in order to evaluate the efficacy of the individual treatments for management or controlling the diseases. And among that, only 3 chemicals found superior, and those were included in the integrated disease management. And among the different management, D6 management found better for the plant diseases or reducing the disease incidence. So this is the overall field out. And from here, the present study revealed that rhizome rot ginger is an important complex disease which is caused by different pathogens like pythium, fusarium, sclerotium, ralstonia, and melodogyna. Even though individual pathogens are capable for causing the diseases, when they are come in interaction, it causes higher percentage disease incidence as well as rhizome rot. And nematode acts as a predisposing factor that increases the severity. Among the different varieties, only one variety found partially resistance, and the external application of elicitors significantly increased the efficacy, increased the expression of different elicitors genes which is involved in the expression, and it is activated in early, middle, and late intervals, which will be helpful for the bacterial as well as fungal infection. And among them, the different elicitors, methyl jasmonate found important. And finally, we have come up with an integrated disease management module, which we can use in the future for the management of rhizome rot disease in case of ginger. Thank you.
Thank you, Archana. Now I will call upon Deshraj Sri Bharti.
He may not be there. Let us wind up the session, sir.
Deshraj is not present.
No information from him.
Okay. So I will conclude the session by saying few words about this one. So in the morning session, 7 candidates were competed for the PR Verma Awards competition for MSc student, and in afternoon session, out of 5 candidates, 4 candidates have presented their research for the PR Verma Awards competition for PhD. So I will thank to the organizing secretary for giving me opportunity to work as chairman for this session. I am also thankful to my co-chairman, Dr. Manoranjitam SK, and the rapporteurs, Dr. Prabhat Kumar and Manjunath Reddy for supporting in this session. Thank you, one and all.
Sir, Archana has taken 14 minutes sir, to complete.
Pardon? Sir?
She has taken 14 minutes to complete her presentation.
Yeah, Archana has taken 14 or 15 minutes. Sir. 14 minutes. So
14.
Yeah, 14 minutes she has taken for completing.
Okay.
Okay, sir.
Sir, if Madam she wants to say anything. Co-chairman.
Yeah. Co-chairman, Madam.
First of all, I want to thank the ISMPP Society for encouraging the students by instituting these awards. And the work carried out by the students, both MSc as well as PhD, need to be appreciated. And most of the presentations are really good. And I feel there is a tough competition. And I thank the organizing secretary, R.K. Mesta, for opting me as co-chairperson of this session. And all the best for the competitors. Thank you, sir.
Now, I would like to thank the chairman, co-chairman, and the rapporteurs for leading the session. Also, I would like to congratulate and thank the participants who have enthusiastically participated in the session. Now, Let's move on to the third and final session of the day, that is on New Horizons in Biological Management of Plant Disease. This session is chaired by Dr. Tamaya N., Dean, College of Horticulture at Babi, who has brought about many outstanding contributions in the field of plant pathology. He has published 98 full-length research papers in referred journals as well as many abstracts and popular articles. He is credited with the development of many technologies and release of crop varieties. He is also the recipient of many awards and recognitions. And this session is co-chaired by Dr. Pranab Datta, Associate Professor at School of Crop Protection, College of Postgraduate Studies in Agriculture Sciences, Central Agriculture University, Meghalaya. He has been actively involved in teaching, research and extension for last 2 decades and has published more than 100 research papers and popular articles. He is a recipient of 27 awards and has developed 10 biocontrol formulations. The rapporteurs for the session are Dr. Sudarshan GK, Assistant Professor of Plant Pathology, College of Horticulture, Mysore, and Dr. Abdul Karim, Assistant Professor of Plant Pathology, College of Horticulture, Sirsi. Now, I invite the chairperson, Dr. Tamaya N., to take over the session. Sir, please.
Thank you, madam, for the nice introduction. I welcome you. Good afternoon. I welcome you all for today's session. That is the New Horizons in Biological Management of Plant Disease. I welcome co-chairman Dr. Pranab Datta, Associate Professor, School of Crop Protection, College of Postgraduate Studies in Agriculture Sciences, Central Agriculture University, Meghalaya. I also welcome our rapporteurs, Dr. Sudarshan GK, Assistant Professor of Plant Pathology, College of Horticulture, Mysore, Dr. Abdul Karim, Assistant Professor of Plant Pathology, CUH Sirsi. Dr. Saab?
Where is Dr. Pranab Datta? I'm Kajal Bisha speaking.
Yes, sir.
Where is Dr. Pranab Datta?
Yes, sir. Good afternoon. Good afternoon, sir.
Good afternoon, sir.
Not video. This video is not on.
No, now my video is on, sir.
Yes,
No,
yes.
Dr.
It's you, sir.
Pranab Datta. Okay, go ahead.
Sir, Chairman, sir.
Yes, sir.
Please
Sir,
proceed.
yeah. Good afternoon. I welcome you all for today's session, New Horizons in Biological Management of Plant Diseases. We have two lead presentation by Dr. Rashmi Agarwal and Dr. S. Nakiran, and two oral presentations. First, I would like to introduce Dr. Rashmi Agarwal, fellow, elected 2019. She was born at Jalandhar, Punjab on 22nd August 1960, educated at Delhi University, and presently working as a head division of plant pathology, ICR Indian Agriculture Research Institute, New Delhi, from 2014 to till date. Madam received many awards, honors from different institutes and also the societies and different organizations, such as Professor M. Wajid Khan Memorial Award by Aligarh Muslim University for outstanding contribution in Department of Plant Pathology in 2018. The second best award, Nanaji Deshmukh ICR Award for Outstanding Interdisciplinary Team Research in 2018. Outlook Agriculture Conclave and Innovation Award in the category that is Innovation in Crop Protection through Scientific Process in 2018 by University Union Minister of Agriculture and Farmers Welfare. And also one of the best award that FAO Fellow to the University of Western Australia, Netherlands in 1999. And also received the National Fellow Award in 2005 ICR, VP Gokhale Award in 2002, Sharda Memorial Award in 1990, Indian Pathological Society Gold Medal Award in 1996, Professor MJ Narasimhan Medal Award in 1997, and also received the KPY Menon Best Poster Award, and also received the KC Mehta and Manoranjan Mitra Award, that is IPS. She also worked as a journal president, IPS, and senior editor and editor, IPS, from 2010 to 2016. And also worked on various aspects, that is the includes fungal pathology, host pathogen interaction, management of plant diseases, and pathogenomics. On behalf of the society and myself, heartily congratulations for outstanding contribution in the field of the plant pathology. Madam, thank you. I request Dr.
Okay.
Rashmi
Yes, sir.
to deliver the lecture on. Yes, madam.
Yes.
Please, madam. Please, madam.
Can you all hear me? Good afternoon, everyone. Can you hear me? Okay.
Yes, madam, you can
Yeah,
hear.
first of all, I would like to profusely thank organizing secretary, Dr. Raghavendra Mesta, inviting me to share with you some of our work on recent advances on biocontrol mechanism through genomics approaches. And I would like to share the slides first of all. Okay, can you see the slides?
Yes,
This
madam.
is okay. This is the topic which I have just selected based on our recent work on genomics and metabolomics of biocontrol agents. And my talk will be divided into basically two major parts. One, where we stand in terms of our literature, and second, what we have done recently in harnessing the full potential of biocontrol agent Chitomium globosum. So as all of you are aware that with the recent omics technologies, which is a holistic view of seeing a molecule within a cell or tissue or an organism, taking all the recent platforms of full genome sequencing, then transcriptomics data, proteomics data, and proteomics, of course, leading to the functional and also the metabolomics. So these are the major tools and techniques in our hand together, which can give us lot of dividends. And these are all aimed primarily at universal detection of first the genes, which we can take from the genomics part, then the expressed genes from the transcriptomics part, and the proteins which are translated through our messenger RNA that can be seen through proteomics. And then finally, what are the different metabolites being produced. So you are all aware that nowadays for eco-friendly management, reducing the pesticide usage and their adverse effect to the environment, there are a lot of studies are there on biological control of plant diseases. And if you look into it, and we have seen that in literature also, and in at country and outside the country, there are a lot of products are available which can be utilized under the field conditions. Still, we many times see that there is inconsistency in our applications, because efficacy is not uniform year to year, and this may be the reason that many of the products, they lose interest by the stakeholders. So if we have understood the whole mechanism of it, then targeted approaches can be taken up for many studies and many leads in the biocontrol through terms of mechanisms. So if you look into the omics, there are different platforms are there, and even if you have generated data on based on the methodologies or technologies available, which I just mentioned, genomics, transcriptomics, proteomics, or metabolomics, but only generating data does not lead us to anything unless we take all the analysis part through using bioinformatic tools. The data generation has to be annotated, then it has to be validated, and then we have to integrate this data, and also we have to annotate to pinpoint some of the potential genes which can be further harnessed, or potential metabolites produced by those genes or gene clusters. So one by one, I'll just take up. The genomics, it includes a study of structure, function, and expression of all the genes in an organism. So this is basically divided into three major categories. So this genomics data helps in looking into a large information. We can understand the genome size. We can also do the sequencing and see how the genome is organized, and how the genes are placed, and how the genome is evolutionary replaced. So a lot of studies can be done if we generate data on the genomics part. So these three things which I have already said, structural genomics, we can come to know the functional genomics, understanding the functions of genes by which are encoded by the genomic sequencing, and then those genes can be shortlisted and then functionally validated through first through expression analysis and then validation through raising the mutants. Then the comparative genomics also gives us lot of information. For example, if you have more than one strain of a particular species, maybe it's a pathogen or a biocontrol agent, particularly today we are talking in terms of biocontrol agent. If two strains are there and you have the genome sequence or the transcriptome data available, the comparative genomics can give you a lot of information how one strain is different from the other strains and what are the genes which are lacking, how much is the redundant genes, and what are the functional genes which are there. So comparative genomics is a very integral part of the whole genomics study. So these are some of the techniques which one can use. I said genome sequencing, then assembly and annotation. These three very important steps are there in analyzing the genomes. And if you look into the application parts of biocontrol agents, you have to first identify genome structures of the strains. It can help us in featuring that. Then also identifying and characterize the genes which are potentially involved in only biocontrol mechanisms. And then characterizing these genes or gene clusters with unknown functions. So it's a very good tool for gene mining. Then you also, I earlier said, they're comparing the genome with other strains. Either can be of the same species or it can be with the related species. So comparative genomics is very important. Then this study also permits us to understand the transcription level. You can have global transcription instead of what we initially used to have the targeted gene approach, fishing out some of the genes and doing only the expression analysis. So here, if we generate the transcriptome data, so we can have the global expression profile of a particular biocontrol agent, either without the presence of the pathogen or with the presence of the pathogen or with the tritrophic interactions, even with the presence of the host. So if you look into the history of this whole genome sequencing related to biocontrol agents, it's not very old. We find Pseudomonas protegens PF5 to be the first MBCA, the microbial biocontrol agent, wherein the full genome sequence was available in 2005. Subsequently, Bacillus amyloleucophaciens in 2007. And then subsequently 2011 onwards, we also started generating data on Trichoderma species, Trichoderma atroviridae, virens, hematum. And Mukherjee's paper 2013 is on the comparative genome analysis. And in literature, we recently found one of the Chaetomium globosum isolate the whole genome sequence available very recently. This is not our sequence, but we have also sequenced one of our own potential strain of Chaetomium globosum and also generated transcriptome data, which I will take up in subsequent slides. So this is a summary which we have taken from the literature only, that how most of the prokaryotes are there for which we have the genome sequence available, because genome size is very small for the prokaryotes, and what are all different highlights of the genome sequence, that what are all genes and what functions they are doing. This is available in the literature for mostly bacterial biocontrol strains. And little information is also there for the fungal biocontrol strains. So this is only the summary depicting what are all different biocontrol strains which have been sequenced, and the draft genome sequences are available in the public database. Further, transcriptomics is another approach, and wherein we generate the data on the whole set of RNAs, including messenger RNA, ribosomal RNA, transfer RNA, and other non-coding RNAs. So whole transcribed genome can be studied, and we can fish out the potential genes if we generate this data. And these transcriptome studies of many of the microbial biocontrol agents have been carried out, and where now from the earlier gene targeted approach, we have not got the whole transcriptome data generated. And from that, we can shortlist some of the genes which are of our interest and can be through analyzed further through real-time PCR. For fungal biocontrol agents, many of the cell wall degrading enzymes or the genes responsible for production of these enzymes like glucanases, chitinases, proteases have extensively been studied. There are a lot of review articles are there. And then recent high throughput sequencing of cDNA. That's what is total RNA-seq data can be generated. This is through high throughput technologies. This has really revolutionized. In addition to the whole genome sequence, the RNA sequence analysis has revolutionized our studies in the even biocontrol agents. So now we have a lot of databases available and which can be further utilized for our useful purposes. So this is in organogrammic form, how the transcriptomic approaches can be taken. It can be through the whole genome transcriptome analysis or targeted genome transcriptome analysis. And you can have all these different techniques, sequence-based, or you can do northern blot, dot blot, or real-time PCR, and also the earlier known hybridization-based techniques, and then which leads to the identification of expressed genes or the ESTs. And then you can prepare library, and out of those libraries, you can harness the either uncharacterized genes or the hypothetical genes to which you can assign functions, or already characterized genes in some other strains which you can utilize in your particular strain to get the information. So these the transcriptome data which is available in the literature, this has been just summarized here. And some fungi transcriptome data also you will find here, like Trichoderma hertzianum T34, very potential strain, well studied in the literature. Then another strain of Trichoderma hertzianum, and these are 3 strains of Trichoderma hertzianum which have been studied. So one paper, very interesting paper I have just gone through, and this is Alatanosova et al. 2013. They have performed a comparative analysis of Trichoderma virens strains and producing this glioviridin or gliotoxin. And they have done this analysis to see what are all genes which are up or down regulated either before coming in contact or at the time of contact, and also after the contact with the pathogens. If you see here, the these arrows indicate all those genes which are upregulated before coming in contact with the pathogen, and yellow ones, you don't see here the yellow ones, you see here the yellow ones, glyk K and glyp P. These two are the potential genes which are upregulated when coming at contact with the pathogen, and some of these genes which are our green. You can see here, this is green, which is upregulated only when after it has come in contact with the pathogen. So these are some of the potential genes which they have done it, that they are involved in mycoparasitic action against Rhizotonia solani. So if you look into the literature, you will find not much information available in terms of these expressions, particularly for biocontrol agents. Although for pathogens and pathogenomics and many of the expressions of pathogenicity related genes or virulence related genes, you will find the literature is full. But for biocontrol agents, not much. So here begins our story on Chitomium globosum. Many of you may be knowing that under National Fellow Project, I was working starting from 2005 onwards on Chitomium globosum. And we did lot of in vitro, in vivo, under field conditions. Every work we have already done so much of work, except for the recent data we generated on transcriptomics and genomics of Chitomium globosum. So the background information I'll just share with you with this slide, that it had strainal differences. You can see in this particular slide CG2 and CG1 and CG6. This had mycoparasitic activity, and CG2 and 3 and 4 and 5, they were total causing antibiosis, although all were Chitomium globosum only. So they were strainal differences. So we try to work out the transcriptomics, and this is a workflow how we worked on the transcriptomics data generation on CG2, which is called causing antibiosis. And our all the results have indicated that CG2 is highly potential in managing a large number of diseases. And the most potential disease was spot blotch of wheat caused by Bibularis sorokiniana. So we developed formulations and we observed its bioefficacy, and also against late blight of potato and wheat rust, and also Escochita blight. So these are all the studies which were earlier done. So we wanted to see how CG2, in what form it is, what are all the potential genes it is producing, the unique genes or the common genes when it is coming in interaction with Bipolaris sorokiniana, the most virulent strain. So we did all these studies, first in vitro, and then we took up the mycelium of CG2 alone, and also in interaction with BS112 and also BS112 separately. So we extracted the RNA, total RNA, and then the messenger RNA was particularly through cDNA synthesis kit, we took out the messenger RNA, and then we gave it for sequencing outside. We didn't do it here. So this is only the raw data indicating how good the data was. And these are the three data sets which were generated. So this data set indicated that this was the sequencing was done through Illumina HiSeq platform. And this generated around 20 to 22 million reads, and the GC content varied from 50 to 58%. Content. And if we did the annotation and plotting of all the genes, we found that roughly nearing 50% of the genes were all matching with Chitomium globosum, and then followed by Mycelophthora thermophila, and then Thalavia terrestris. So if you see here, this is the sequence homology which we got with other related species, most of them belonging to Escomycota. So this is the volcano plot which we generated on the differentially expressed genes. So the green zone indicates that these are all highly significant DEGs. These were expressed differentially. So these were the genes which further can be harnessed for fishing out some of the potential genes. And if you see these two graphs, you find that this is the genes which are only in CG2 control without coming in contact with the pathogen. And these are in the interaction. CG2, when it came in interaction with BS112 pathogen, and these are all differential vein diagrams which we have, all permutation combinations were done to analyze the data. So the non-overlapping regions are the unique genes, and then overlapping regions shows the common genes which were expressed during this transcriptome analysis. Further, when we did the geoterms analysis, these all functionally, they were categorized into three categories: biological processes, cellular components, and molecular processes. And this is very significant analysis for the pathways. So we found that the most of the genes, maximum genes, were involved in metabolic pathways. You see here 1062 genes were in the interaction when they were in interaction with BS112. So, and also followed by biosynthesis of secondary metabolites and biosynthesis of antibiotics. So these, some of these were very important genes which can further be utilized, including and many other genes which included chitinases, proteases, and then polyketides and antimicrobial peptides and so on. So this is a list of the potential genes which have been classified based on the functions. These are the genes involved in cell wall degradation. These are the genes involved in secondary metabolite biosynthesis and antibiotic production. And these are some of the transporter genes, transcription factor genes, and other antagonistic related genes, which we annotated through our bioinformatics tools. And these are some of the pathways which we could decipher. All these blue ones are the colors which are indicating the genes which are upregulated, and the red ones are all the downregulated genes during this particular pathway. So out of these genes, some of the genes were shortlisted for expression analysis at different time intervals. And if you see here, during interaction, most of these genes are all upregulated, either at 4 days post-inoculation or 8 or 12 days, three time points we took. And you will see many of these are very important genes, proteases, then cyanate hydrases, and superoxide dismutases, and one is this protein kinase domains. So these are some of the very potential genes which are getting upregulated when they are coming in contact with BS112. Individually, if CG2 alone is there, so it is not getting expressed. So it means these BS112 is able to induce some of these genes and activate the pathways for production of many of the metabolites. And this paper has been published in Frontiers in Microbiology in December 2020. So next was the proteomics approach that also gives us very much related to all the proteins which in a particular species, which are present at a particular time at particular environmental conditions. So these are also very essential for the biocontrol mechanism and the process. You can even do the reverse genetics and find out what are all the proteins through this approach. And then you can go back and see what are the genes which are producing these proteins. So these are very different applications one can have. One can have the proteome fingerprint of the strains, like we have transcriptome fingerprint, and then you can also go for differential expressed proteins during biocontrol process. So we did it for Chitomium globosum, and this is comparative. Okay, so you can see this is a 2D gel proteins of our CG strains, and we did all the analysis and identified many of these genes through MALDI-TOF LCMS. And then one protein we could go back to, as I mentioned, go back to the genes which are, this is a list of all the proteins which were identified in Chitomium globosum through our study. And one of these heat shock protein, which we characterized by our studies, that I will take up in after 1 or 2 slides. And this is only summary of the proteomics study which I'm mentioning here. Since our database was not so strong at that time in terms of proteomics, we got 79% of the proteins were all categorized into hypothetical proteins, which are all uncharacterized proteins, and 21% of the Chitomium globosum proteins were all earlier characterized. So if you further categorize these 21% proteins, so 24% out of these was all matching with Chitomium globosum, and rest with other genera belonging to Ascomycetes, including Aspergillus species. So out of that one protein, which I said the heat shock protein, we could take out the gene for that, and we could analyze this in Chitomium globosum isolates, and we found that it is present only in Chitomium globosum, not in other Chitomium species. And we also expressed it, this particular gene, in PET17 expression vector, and with the protein which was the gene which we did the phylogenetic analysis. And if you see, this is our HSP small heat shock protein, and this is with other existing database. So it was matching almost 100% with the existing database heat shock protein. So this is particular small heat shock protein, which we characterized to in Chitomium globosum, and this has had the tolerance to temperature and also to the salt tolerance. So this is another study which we could understand that if biocontrol agent has tolerance to the heat and also to many of the stresses in the environment where we are applying, so this has a potential. It further supports the bioefficacy of the biocontrol agent. So this was the metabolomics which we carried out with the same set of experiment, like CG producing metabolites either alone or in the presence of our pathogen. So if you see here, some of these dodecanes and tetradodecanes, alkenes and alkanes, these were the potential compounds which we could identify. They were being produced. Some of the unique compounds were being produced only in the presence of the pathogen. So this is unique compounds produced only in the presence of pathogen, and these are other compounds which were produced in the absence of the pathogen by Chitomium globosum potential strain. So some of these antifungal metabolites which have been identified are listed here, and these have properties for antagonism. And if you see the classification of these compounds, alkanes and alkenes were the major compounds, followed by ketones and then alcohols and aldehydes. So these secondary metabolites were expressed only during interaction. If you see here, these green ones. So these are very potential secondary metabolites expressed only when it is interacting with the pathogen. So this is all about our work on CG, and most of the work is published, and still some more needs to be further validated. So if I summarize my talk in this post-omic era, what can be our leads in this and how we can harness the potential. So the genome sequence of all the microbial resources, which are mainly utilized for the biocontrol, this is backbone. It's a holistic backbone to support further studies and developing methodologies and formulations. And then this omics approach also helps us in understanding the complexity of the modes. We have seen in the literature that a single biocontrol agent doesn't function in a single manner. If a biocontrol agent is mycoparasitic, it also produces secondary metabolites and many other functions. It's also competitive and also induces resistance and brings about growth promotion. So multi activities are being seen, and so it's a very complex. So some of these techniques can help us in unraveling the complexity. And also the genetic distance and you know that the question many times arises when we are going for registration, that it should be very safe. So this genomics and transcriptomic data further supports that it is safe when we do not have genes which are involved in pathogenicity to the humans. So these are some of the leads one can get through generating the genomics and transcriptomic data, and also proteomic studies, which are directly related to the functionality of the biocontrol agents. So the protein effectors also can be identified, and if we are able to identify the effectors, we will be able to identify their targets in the pathogen and also in the host plant where we are applying. So it will give us a lot of clues for tritrophic interactions. So that is a important support for the proper application of bioformulations under field conditions. So with these brief presentation, I will end my talk here, and thank you all of you for patience hearing, and protecting our plants will lead to protecting the life on this earth. Thank you, all of you.
Madam, thank you for nice presentation with valuable information. I request the audience, if any clarification, you can ask. Hello?
Yes.
If any clarification. If you don't have any clarification, we'll move on to the second topic by Dr. S. Nakiran, endophytes and its application in plant disease management. I will call upon Dr. S. Nakiran. Before that, I will introduce the Dr. S. Nakiran. He was the elected fellow 2009. He was born at Singapunari, Sivagangai district, Tamil Nadu. He completed BSc and MSc and PhD at Tamil Nadu Agriculture University, Coimbatore. Presently working as Associate Professor of Plant Pathology, Department of Plant Pathology, TNEO, Coimbatore, from 2009 to till date. Previously served as an Associate Professor in KVK Villapuram from 2006 to 2009. Postdoctoral Fellow at University of Manitoba, Canada, from 2003 to 2004. And also visiting scientist to South Korea in 2002. Earlier, he was worked at different research stations and also the other institute.
Sharing problem.
Dr. S. Nakiran received the following informations and also they received the awards. Hello. He received the following awards. Best Presentation Award, IRI, in 2005. Second Best Award, OM Lakshminarayan Reddy Shield and Medal Award in 2005. And PR Verma Award in 2004. is mainly concentrated on biological control of plant diseases, biotechnology of trichoderma, PGPR, molecular plant pathology, oyster and milky mushroom, optimization of delivery system under precision farming. Now I will call upon Dr. S. Nakiran is going to deliver the lecture on endophytes and its application in plant disease management. Please. Dr. Nakiran, sir.
I think. You.
Sir, please share the slide. I mean, presentation.
Yeah.
Slideshow option.
Yeah. Click the slideshow option.
Right.
Can you? Okay.
Now visible.
Okay. Thank you. So, respected chairman, co-chairman, and the rapporteurs, and the organizing secretary of this great day of this national symposium. So at the outset, I would like to thank the organizers for giving me this opportunity to share my experience on does bacterial endophytes contribute for the management of plant pathogens. So this is the question which is brought in front of our mind. So when we think of that, so what is the status of global survey on microbials? What is that which is want to present in front of us? So now if you see the investment in the case of the microbial discovery and development, if we analyze, there is a chances for more than $11 billion to be invested, which contribute to around 60% of the market share at a global level. And if you see the microbial products, they grow at a very faster growth rate. And because of that, number of multinationals are investing on this. So considering this scenario, if we analyze what is the number of biologicals that has been registered with the Central Insecticide Board in India, if you analyze, at present, if you see, there are only Trichoderma viride, Trichoderma harshianum, Pseudomonas fluorescens, and Bacillus subtilis alone. So these are the 4 major products which are found to be registered with the CIB in India. But the wide opportunities are found to be more. So considering this, let us go what to discover and how the diversity of the endomicrobiome can be utilized. In most of the cases, if we peruse the literature for the past 3 or 4 decades, so the work has been continuously, they have done on the exploration of rhizosphere microflora. But at the same time, the work on the exploration on the diversity of the endomicrobiome has been only under limited scale. So if so, how to explore the endomicrobiome? So who are they and what they are going to do? So based on this, the journey on the bacterial endophytes, if we talk, there are diverse nature of endomicrobiome present in the plant community. So these can be used for the management of fungal pathogens, bacterial pathogens, viral pathogens. So next, if we see, why can't we explore, if there is a potential, why can't we explore these endophytes with a broad spectrum of action? So these endophytes, if you see, the endophytes with antifungal action, antibacterial activity, and antiviral nature, so they can be used for exploring the in-plantar resistance. So how? The journey to discover the innate immunity response against fungal pathogens of global significance. So how? Now, let's see how the journey can be we have made for the exploration of the endophytes for the management of the fungal pathogen. Let us take the Phytophthora infestans. So this is one of the globally important fungal pathogen in the world. So this is a destructive necrotrophic pathogen, and this Phytophthora infestans they have the isolated from Bichu, Laching, and Nilgiris from different locations and even from Daman. And these Phytophthora infestans has been confirmed through different means. One, through phenotypic observation of the morphology of the pathogen by observing the hypha, and they have possess the lemon-shaped sporangia, the semi-papillate sporangial structures, and after the phenotypic confirmation, it has been confirmed through molecular characterization using the internal transit spacer, RXLR gene, AUR3A, beta-tubulin, G protein beta subunit, and 1,3-beta glucanase. So these are the aseptate nature of the hypha. This is the lemon-shaped papilla which has been seen. And when we observe these sporangia and through scanning electron microscopy, so we can see the presence of the sporangia which has found to be present on the leaf surface. And this is the stage-wise release. So the sporangia, encystment of the protoplasm into zoospore takes place, release of the protoplasm, release of the zoospores occurs, and it gets emptied over here. So this, when it comes in interaction with that of the host surface, the zoospore germinate, they gain entry through that of the stomatal opening and causes infection. Then after that, after confirming the identity of the pathogen, next we were interested in why or how does this endosphere engineering with the bacterial antagonist can mitigate late blight of potato. So how best we can challenge the phylloplane? So here, the endosphere, from the endosphere region, all the endomicrobiome has been screened, it has been sieved and dissected, and based on the endomicrobiome engineering, so the effective endomicrobiome after screening, they have been applied to the phyllosphere, and how best it is going to interact with that of the pathogenic organism, the Phytophthora infestans. And during the interaction, what are the things that goes on and how it results in suppression of the plant pathogen. So these were going to be investigated, which will really come out with a sustainable management strategy through man-triggered immunity. So for which we have isolated this bacterial endophyte from Sikkim, from Shimoga, Kodaikanal, Nilgiris, Theni, and Ramsamutram from different solanaceous crops and from the potato. So out of which we have identified 644 bacterial endophytes has been isolated. And after the isolation, we can categorize what are those organisms. So the organisms were isolated from the leaf, flower, stem, root, and from various different plant parts. So the total diversity, if you analyze, after the molecular characterization, there was around 12% of the bacteria, they belong to proteobacteria, 10% were distributed as Bacteroides, and 80% belong to Firmicutes. So this is the distribution pattern of the bacterial diversity in the endosphere. And what is the antagonistic potential of those 644 antagonists against late blight of potato? So that is one of the next question which is found to be present in front of us. So all the isolates were found to be screened, out of which most of the isolates, so from 644 isolate, we have narrowed down to 48. From 48, we narrowed down to 10 isolates, which has more than 90 to 100% inhibition of Phytophthora infestans. So here, if you see this photograph where there's a 100% suppression of the pathogen over here. And likewise, everywhere, if you see, the biofilm formation is found to be best in one strain. In one strain, it produces more of antibiotic and inhibition zone can be observed. And here, as per the earlier speaker, as Dr. Rashmi Agarwal was also saying, so a multiple different mode of action is found to be coordinating by a single organism to counter the suppression of the plant pathogenic organism. So these are the different isolates which are found to be performing better for the suppression of the plant pathogenic organism, the Phytophthora infestans. So here if you see, so this is the growth where we can see at this particular point of interaction, the pathogen does not develop here, it does not grow at all. So here there came a question, so what might be the metabolite that has been distributed in this region in the presence of the Phytophthora? And what are the metabolites that has been distributed or that has been secreted by the pathogen in the absence of an antagonist? And what are the metabolites that has been synthesized and secreted into the media by the bacteria alone in the absence of a pathogen? So a monotrophic and ditrophic interactions, how does it reflect on the diversity, on the profile of the metabolomic thing? So that was investigated. So here, if you see, the next question that we have is, does the endomicrobiome inhibit the late blight of potato? If so, how best these bacterial endophytes can inhibit? So here, at the point of the ditrophic, that is in the point of interaction, from the ditrophic interaction zone, where when we can able to see and perfect inhibition zone of the growth of the pathogen. So the agar from this region was excised. It was subjected for the extraction of the compounds that were found to be secreted into the agar media, which comprise of both volatile and non-volatile compounds. And those compounds were found to be identified as 3-deoxydeaminoic acetone, phosphorothioic acid, glucuronamide, clindamycin, ethanamine, benzethonium, then streptovitamin. So like that, an array of compounds was found to be produced during the ditrophic interaction alone, which were not found to be produced during the monotrophic interaction by the pathogen alone. Whereas that was found to be one which was found to be very, very specifically induced during the interaction of the pathogen and biocontrol agent. So those compounds are found to be giving a clue to the next level why they may not be the chemical or the biomolecule responsible for the suppression of the plant pathogen. And apart from that, if a biocontrol organism or if an endophyte, if it want to be effective, the formation of the biofilm is very, very essential criteria. Unless and otherwise the biofilm formation is not taking place, then the biocontrol organism is not going to serve the purpose. Unless so, because of that, the most of the effective bacteria we checked, and all these were grown in the biofilm production medium, and all the organisms were found to be differing in their ability to produce the biofilm, and most of the organisms was found to be performing better in production of the biofilm. So based on the antimicrobial peptide, the next mode of action is the presence for the detection of the antimicrobial peptides. So the antimicrobial peptides comprising of 13 different antimicrobial peptides, including iturin, surfactin, fengicin, flipastatin, bacillomycin, fengicin. All these antimicrobial substances or the antimicrobial peptides were found to be assessed through PCR mechanism, through molecular characterization, where most of the isolates, they were having the capability to produce the antimicrobial peptides. But among those, the isolates NM297 and these isolates were found to be producing the more number of antimicrobial peptides, and based on that, we have picked out 10 different effective organisms which has a cumulative performance of different mode of action to challenge the phylloplane area. So here we engineered the phylloplane with that of the endophyte. So here the colonization of the phylloplane by the effective bacterial endophyte. So how do they colonize and how do they multiply and how do they interact with that of the pathogen in the presence of the bacteria? So that was investigated. So based on the investigation, to our surprise, we were able to understand the hyperparasitic nature of Bacillus amelia isolate 345 on Phytophthora infestans, sporangia, and mycelium. So here, if you see, we can see the beautiful colonization of the bacterial cells on the sporangia of the Phytophthora, and it formed a continuous layer on the sporangial surface. And after that, if you see, it cleaves the sporangial wall and it leads to the multiplication of the bacterial cells inside the sporangia, leading to lysis of the sporangia. And not only that, the hyphal cells were also completely colonized by the bacteria. And you just kindly imagine at 5000x magnification at 10 micrometer distance, a huge number of millions of bacterial cells are found to be there. So to such an extent, they are found to be competitive enough to colonize this particular organism. So this is how the bacteria colonizes the mycelium on the hypha, and it also results in the complete colonization of the sporangial wall. So like that, all the different 10 effective isolates were also found to be performing equally effective in colonization of the sporangia. So this is the another photograph which has been taken for another isolate of Bacillus amelia, where there was a complete colonization of the sporangia, and followed by cleaving of the sporangia was found to be observed. And this, beyond doubt, it clear that the organism of our interest was having the capability to grow on the sporangial wall and to lyse the sporangia. And this is the opening and the release, the point of the release of the zoospores from the sporangia, where the bacteria Bacillus willisensis and another effective organism which was colonizing the sporangia, and it would completely ramify the sporangial surface, resulting in suppression of the pathogenic organism. So these are the 10 different bacterial endophytes which we have identified, which is found to be having the capacity to inhibit the pathogen to an extent of 90 to 100%, and the identified organisms were referred as Bacillus ameloications, Bacillus willisensis, Bacillus subtilis subspecies speciesini, Bacillus subtilis subspecies subtilis, then Bacillus safensis, Bacillus subtilis subspecies stercoris, and all these particular organisms were found to be effective, and they have been developed as an EC formulations, and it is has been applied for the phylloplane and for the plant surface for the management, which is also found to be under evaluation in the field trial. So totally, if we examine what are the various different metabolites that has been produced, and if you map the KEGG pathway, so the mapping of the KEGG pathway revealed that the electron transport cycle, so the different cycles, if you see the TCA cycle is getting activated, and the TCA cycle gets activated, the electron transport chain, oxidative phosphorylation, aspartate pathway, then ethylene pathway, citric acid pathway, terpenoid backbone pathway, fatty acid biosynthesis pathway, then beta oxidation, alpha linolenic acid conversion, then biosynthesis of unsaturated fatty acid glycolysis, and all these different pathways are found to be triggered simultaneously during the ditrophic interaction by the bacterial biocontrol agent, the endophyte, and which results in production of several molecules which acts as a precursor for another pathway and results in the suppression of the plant pathogen. So this is a classical example that we have discussed about our work on late beta potato. In addition, our work on journey on how does the viral diseases of the plant viral diseases are found to be suppressed by the bacterial antagonists. So one of the major problem in cotton is in South India is the tobacco streak virus, which belongs to LR virus group. And here what we did is from the resistant genotypes, we have isolated the bacterial endophytes associated with that of the cotton plant. And those plants were having resistance towards the establishment of tobacco streak virus. So these are the array of different metabolites, different bacteria which has been present in the resistant genotypes. So here the distribution pattern, if you see, the distribution of the endophytic bacterial community was found to be comparatively, that is a Bacillus endophyticus, Bacillus ameloications, Bacillus subtilis, Bacillus licheniformis was found to be noticed in the case of the resistant, whereas in case of susceptible genotype, Bacillus cereus, Bacillus willisensis, and the Enterobacter. So this was only noticed. So the diversity is found to be more in resistant genotype compared to that of the susceptible genotype. So considering that, we went for doing another experiment of Bacillus ameloications, subtilis megatherium, licheniformis, Bacillus methylotrophicus, and Bacillus ameloications. So these isolates were sprayed onto the phylloplane, and how best they are going to interact with that of the virus and how it going to suppress. So this particular work we have published in Frontiers in Microbiology in 2017. And here if you see, simultaneous inoculation, so pre-inoculation of the virus challenging with that of the bacteria, then simultaneous inoculation of the bacteria combined with that of the virus, and post-inoculation spray. So when we compare the different methods of application, the simultaneous application of Bacillus ameloications VB7 and Bacillus licheniformis completely reduce the lesion formation of tobacco streak virus in the local lesion host cowPC 152. So what does it indicate? So why the lesion gets reduced here? So this is how the complete reduction of the lesion was observed, whereas in the inoculated control, we can see more than 200 lesions were found to be noticed in the case of the inoculated control. Likewise, if you see the in vitro efficacy of the Bacillus in the reduction of the symptom expression in copy here, the effect, if you see the T4 and T5, so that is the challenging of the plant with that of Bacillus licheniformis is T4, and Bacillus ameloications is the treatment 5. So the pre-inoculation, simultaneous inoculation, and post-inoculation, all three different methods of inoculation, the number of lesions by the tobacco streak virus was found to be reduced per leaf when it is compared to that of the inoculated control. So when we compare with that of the virus inoculated control, the number of lesions was found to be more than 2 to 3-fold higher number of lesions were found to be observed in this particular case. And not only that, this particular Bacillus species was also used for the management, not only in the local lesion host, we also attempted that in the cotton hybrid RCH659, which was also found to be susceptible for this particular tobacco streak virus. So when we see that the application of baseless amyloid liquefations, foliar application of baseless amyloid liquefations resulted in 20% of tobacco streak virus incidents under the field conditions, whereas in the case of the control, untreated control, there was around 44% to 45% of tobacco streak virus incidents was found to be noticed, which indicate that foliar application under the field level is also found to be having a sustainable effort to suppress the expression of tobacco streak virus. Not only that, in addition to that, it was also found to be playing a vital role in improving the plant height, in improving the number of flowers, and improving the number of bolls, and seed cotton yield was also found to be increased when it is found to be applied with that of baseless amyloid liquefations in this particular case. So boll height, that is number of flowers, number of bolls, and seed cotton yield was also found to be increased. So if so, what is the status of the virus titer in the infected plant when it was challenged with that of the baseless species? So here if you see the baseless amyloid liquefations VB7, in this case if you take, so the virus titer was found to be 0.5 OD value at 405 nanometer, whereas in the case of the control, it was around 2.5. The virus titer was found to be, that is more than 200-fold increase in the virus titer was found to be observed. And the field trial, this was the field trial which we conducted under the field conditions. So ultimately, what we would like to conclude here is, so the foliar application of baseless amyloid liquefations results in triggering the immune response of the cotton plant, where it goes and increase the plant growth promotion, increase the number of flower bolls, seed, cotton yield, and reduce the disease intensity. And this particular work we have published during 2018 in Agriculture, Ecosystem and Environment. And here, what is the thing that we are found to be lacking here is, what is the molecular mechanism behind this suppression? So that was found to be unanswered. So for that, we carried out the same type of studies with that of the another crop also. This is a chayote, which was found to be highly susceptible for the viral infection, that is chayote inhalation yellow mosaic disease. And here the chayote was dipped in baseless amyloid liquefations at 0.5% for 20 minutes, and the 4 seeds were used, chayote seeds were used for planting. So after planting, so the disease, if you observe, it was found to be performing well for the management of the chayote inhalation yellow mosaic disease with bioagent, antiviral principle, where the Mirabilis jalapa root extract was used for the suppression of this particular virus. So the combined application of baseless amyloid liquefations along with that of the foliar spray of root extract of Mirabilis jalapa at fortnightly intervals resulted in complete suppression of the chayote inhalation yellow mosaic virus under field conditions. So why the suppression, and what is the treasure in the genome of VB7? So we were interested to go for genome mining of baseless willisensis. So here when you go for complete genome sequence was done, where the genome length was around 3 MB size, and the GC, that is GC content, was 46.79, and the protein coding sequence was 3090, and number of functional proteins was 2584. So based on the blast that we have made, so earlier, that is this particular baseless amyloid liquefations, when we go for whole genome sequencing, it was identified as baseless willisensis, which was found to be a very, very closely related organisms with that of the species of baseless amyloid liquefations. So baseless amyloid liquefations and baseless willisensis are remaining as a very, very minor differences in the genome organization, and both are found to be same in this particular case of our study. So here, when you analyze the genome organization, we were able to dissect the presence of 7 different regions, which is responsible for the coding of the non-ribosomal polyketate synthase. So the antibiotic biosynthetic genes was found to be present here. So the region 1, which we identified, was NRPS, non-ribosomal polyketate synthase. Region 2 was once again the polyketate synthase, which is responsible for butyrosine A and butyrosine B, whereas the first NRPS was for synthesis responsible for surfactant synthesis, and the region 3, which is the terpene synthesis, which is associated with the synthesis of terpenes, and the region 4 was trans-ATPKS, where polyketate synthase was found to be synthesis here, and the region 5 has trans-ATPKS, which is responsible for the synthesis of difficidin, and NRPS 6 region was responsible for the synthesis of bacteriocin, And the other region 7, it was found to be unidentified what it is having coding for. So it's a non-coding one. And the region 8 is a lanthipeptide. And this is responsible for the synthesis of difficillin. So lanthipeptide is a potential antiviral protein, small molecule, which is responsible for the suppression of the viral infection. So based on the analysis, we were able to see that 14 secondary metabolite clusters were identified from the Bacillus valisensis, and they were responsible for the synthesis of surfactant, basilibactin, bacillicin, difficillin, pengicin, basilein, macrolactin, calimantin, butyrosine, and plantathiazolizin. So all these antibiotic genes were also found to be confirmed through PCR. Also, the presence of the genes were found to be detected. And if we analyze the tritrophic interaction between Bacillus and GBNB and tomato, so here in the earlier we said that it is found to be effective for the management of the tobacco streak virus in cotton. But the question which was found to be there, what is the molecular intricacies which is found to be taking place between the host pathogen and environment? So to understand that, we are looking in for the MAP triggered immunity, the microbe associated molecular pattern triggered immunity. We are going on understanding of all those things. So here, if you see the application of the Bacillus amelioifications, where the number of lesions is getting very much reduced compared to that of the inoculated control. So what is the mechanism of action involved in the suppression? Whether the MAP triggered immunity operates. So for that, we were looking for the flagellin as a MAP molecules, elongation factor, peptidoglycan, lipopolysaccharides, sphalanin, cell wall beta glucan, and cell wall chitin. So these MAP molecules are having the responsibility to trigger MAP triggered immunity or not. So that was found to be investigated here. So is there any regulation of the different genes, and what is its impact on suppression of GBNB? So the agroinfectious clones which confer where the elongation factor and flagellin was constructed, and the agroinfectious clones were added to the rhizosphere region, and it was also spray inoculated onto the leaf surface. So here, this is the one which has been challenged with the GBNB, where the number of lesions are found to be very much reduced, whereas in the case of the inoculated control, the number of lesions and the plant gets severe infection and results in death of the plant in tomato in this case. So if in this case, in the challenging plants, we went for the assessment of the different gene expression. So the mitogen activated protein kinase, and the Ricky transcription factors, then PR proteins, PAL, PPO, ERF, VSP, and PDF 1.2, then that is lipoxygenase and NPR1 gene and JAR1, jasminic acid response gene. So all these defense related genes were found to be investigated, and this is the total overall comparison of the different defense genes real-time PCR. So these were analyzed. So based on the analysis, what we could understand is, in compared to that of the inoculated control, there was a down regulation, whereas in the case of the rest of the cases, where if you see in the case of elongation factor inoculated or flagellin or agroinfectious clone or Bacillus alone, in all the irrespective of the cases, if you see, it was compared also with that of the Bion, where the expression of mitogen activated protein kinase was found to be more. So when the transcription factor is getting increased, the mitogen activated protein kinase gets increased, and next the MIC factor, there is a Ricky transcription factor, Ricky 33B also gets triggered in the case of the challenge inoculation. Next is the NPR1 gene, which is the hub for triggering both ISR and SAR get activated, and followed by the PR1 genes was also found to be activated here. And subsequently, if you take the other gene, the PAL gene, phenylalanine ammonia lyase, get increased over here, and polyphenol oxidase was also found to be triggered, and lipoxygenase gene, which is the key enzyme for triggering the ISR pathways, was also found to be triggered, and jasminic acid responsive gene was also found to be upregulated compared to that of the transcripts, which was found to be downregulated in the case of the inoculated control. And the expression of the MIC2 transcription factor, they were also found to be increased here, and the expression pattern of the PDF 1.2 was also found to be increased. So the constitutive expression or the combined expression of all these different genes might have contributed for the suppression of the virus titer in the plant system. So that might be the whole reason behind the suppression of the viral infection of GBNB and tobacco streak virus and chayote mosaic also. So here, when we inoculate with the agrobacterium mediated MAM clones the VB7 and BTH, the virus titer, what happened when flagellin alone was inoculated, when elongation factor and bacillus and BTH, agrobacterium alone when we inoculate, what is the virus titer? And in inoculated control, the virus titer was checked. But here, and the virus titer in the inoculated control was found to be 1.24, whereas in case of elongation factor and in flagellin agroinfectious clones, it was found to be 4 times lesser than that of the inoculated control. And simultaneously, in the case of bacillus also, it was also equally performing well as that of the agroinfectious clone, where the reduction in virus titer was found to be observed in a different way. And next, not only that, so the coat protein gene transcript, so how it is found to be present in the plant system when it is inoculated. So in the case of the coat protein transcript, if we observe 9th day after inoculation in flagellin, elongation factor, bacillus ameliorations, and all the other cases, the virus titer was found to be up to the 10 to the power 8 in case of inoculated control, whereas in rest of the treatment it was around 10 to the power 5 was found to be noticed in the case of the virus titer, which also beyond doubt, which explains that the virus titer has been also reduced to the extent. So this is the pictorial abstract of the entire finding of why Bacillus valisensis VB7 is found to be performing for the suppression of the virus. So here if you see, simultaneously it results in the activation of both ESR and SIR pathway through this particular mechanism, where the flagellin elongation factor, they combine with that of the pattern recognition receptor. It triggers mitogen activated protein kinase, and mitogen active protein, then transcriptional factor reprogramming takes place at the cellular level, and the defense proteins are found to be triggered in this particular case. Next, the validation of the effective consortium of Bacillus against on the field condition was also carried out, and the field trial was conducted, and the foliar application of this Bacillus valisensis EC formulations was attempted, and it was also found to be performing well in suppression of the disease under the field condition when it is treated as a seed treatment, soil application, and foliar spray. So finally, to conclude, so these particular organisms, what of our interest, the Bacillus valisensis, it was found to be screened best as a Bacillus isolate for against GBNV in cowpea. Then it reduced the number of local lesions through the induction of PAL, PP1, PO. And the MAM genes characterized for the best isolates, where those MAM genes were responsible for triggering and for suppressing the virus titer. And agrobacterium mediated tritrophic interaction assay also confirmed that the virus was found to be reduced due to the application. And qRT-PCR analysis also said that that is reflected that the virus titer was found to be very much reduced, and the complete genome sequence of Bacillus valisensis. And all these things cumulatively come to a conclusion that there's a suppression of GBNV symptom expression under field conditions. So here I would like to confirm that the endophytes, so these, they use multiple different pathways, and one molecule which is coming to us of metabolite, it is used as another product in another pathway, and it leads to simultaneously several other byproducts, and then we come out with a defense products, volatile organic compounds, non-volatile organic compounds, the triggering of the MAM genes, which are found to be responsible for the suppression of the viruses and the fungal pathogens. So this is in short about the what is the secrecy of the endomicrobiome, how it plays a vital role in the suppression of the plant pathogens, that is the necrotrophic pathogen Phytophthora infestans, and in the case of the virals, the tobacco streak virus and GBNV. So this is in short, and this is our team where who are responsible for the build-up of the story up to this level, and we acknowledge the help and the mentor, Dr. V.G. Malathi, for us for coming to up to this level. And I once again thank the organizers for giving me this wonderful opportunity to share our experience on the travel on endophytes and its role in suppression of fungal and viral diseases. Thank you.
Thank you, sir, for excellent presentation. Now the topic is open for discussion. Sir, I have one doubt.
Yes, sir.
What is the mechanisms involved in the reduction of the virus inoculum and also the virus titer?
Yeah. So here in the case that we are talking about the MAM triggered immunity, that is the MAP molecules, they trigger the defense responsive genes. So we, based on our understanding on defense reactions, So more than 8 to 9 different defense-related proteins, which are found to be playing a vital role in ISR and SAR, has been triggered. And this might have resulted in the suppression of the virus titer in an either maybe in a direct or indirect way. So our understanding is the MAM triggered immunity might be responsible for suppressing the virus titer.
If any clarification? Thank you, sir, for excellent presentations. Now I will move on to oral presentation. I will invite Dr. C.S. Vivek Babu. He is going to deliver on molecular insights of plant probiotic fungal endophytes, augmenting forskolin biosynthesis and disease management in coleus. Please, sir. Dr. C.S. Vivek Babu.
Sir, I am on line. Please allow me to share my screen actually from the organizers.
Beautiful presentation, Dr. Nakiran. Beautiful presentation.
Thank you, Doctor.
I enjoyed because you are versatile. Fungal pathology, virology, genetic engineering, biotechnological management, biological management, everything. We are proud of you.
It's all because of your good wishes and friends like you.
Okay.
Sir, can we start?
Go ahead, sir.
Yeah. So good evening, everyone. Myself, Dr. Vivek from CFTRI. Primarily, we will be focusing on molecular insights of plant probiotic fungal endophytes, augmenting forskolin biosynthesis and disease management in medicinal plant of Lycopodium coleus forskolin. So, just I will touch upon because 10 minutes it has been allotted time. Please, I request the chairman wherever you want to interrupt me, sir, please stop me there. So just I will give you a bird's eye view of the work whatever we have carried out in this domain. So basically we all knew that what are these endophytes? Endophytes are which has been plant associated microorganisms which has been residing within the plant tissue, and they are the source of innumerable number of metabolites, and also they have an what you call it as enormous potentiality in triggering the secondary metabolites within the plant system actually. So these are some of the important papers which has been comes out, and then we have seen that there is two groups from Cornell, from Max Planck. They have been observed it on Arabidopsis of the role of these endophytes, that is Coltotrichum, and on this Arabidopsis mock plants, and also in uptake of phosphates in the system actually. So this is general habitat for the general overall audience. There it is why do we call them as endophytes, which has been present within the endosphere tissue of the plant. So basically let us come to the coleus plant itself actually. So we have taken this as a medicinal plant as a model system to understand its plant probiotic nature of the few endophytes, and also in plant enhancement of secondary metabolite, that is a forskolin within the plant system. So in this context we all knew that one forskolin. Yeah.
Sir, your voice is not audible, sir.
Is it clear now?
Sir, can you increase your voice, sir?
So it is all, my audio is full.
Come a bit near to microphone, sir. Your voice is not that audible. It's
is it
low
clear,
pitch.
sir? Is it clear? Is it clear now? Maybe
No, sir, not yet. Be louder.
Yeah. Is it okay? 1 minute, sir. I'll check my audio system. Is it clear?
Okay, but please be loud, sir.
Yeah, sir. Please. Okay. So in that cases, like it is forskolin, we have we call them as a diterpene or labdin diterpene molecule, which has been of economically important because of its two components. Like it has been used in extensively used in glaucoma management and also weight loss. And there are so many companies which has been dependent on these roots for the quality amount of forskolin content within the root system actually. So this is an overall the demand for this forskolin component. And then also, so let us come to directly to the interaction of plant probiotic endophytes in forskolin or like it is coleus forskolin. So I'll restrict my talk for only 3 plant probiotic fungal system which has been innate or inherent fungal system which has been associated with the roots. One is the Fusarium rhodolens and also Phalaenopsis and also Macrophomina. And these 3 which has been isolated from the root system and also one from stem system, which had got a huge impact on the forskolin content, enhancement, in plant and also in the disease management. So these are the generally the source of endophytes which has been isolated from root and leaf and also stem system of forskolin plant. And these are the preliminary screening methods, that is like how does they have an impact on the like it is forskolin content enhancement, and this is generally primary productivity of the plant. And then we have noticed that initially after screening there are 3 organisms which comes out to be very efficient in enhancing in-plant of forskolin content in that actually. So out of these 3 organisms, RF1, SF1 and SF2, and which has been deposited under NCM, National Collection of Industrial Microorganisms. So these 3 which belongs to the class of Fusarium, Phalaenopsis and Macrophomina. So we'll emphasize it as a plant probiotic system, so which has got an impact on even on the field system that we have evaluated. So that is forskolin content which has got a huge impact, like it is even that is a percentage of forskolin which has been almost double the forskolin content, almost, or it is 80% of the forskolin content which has been enhanced within the root system only upon the inoculation of this class of organisms in that individually. So then upon just it's a molecular insights in that actually upon real-time qRT-PCR analysis of the forskolin pathway biosynthetic genes in that. So most of the terpene synthesis which has been responsible for forskolin biosynthesis has been highly upregulated in presence of these organisms or during its interaction. So TPS1 and TPS2 and TPS3 and TPS4, which has been highly committed step in forskolin biosynthesis, melanin oxide biosynthesis, these organisms has been highly interactive during the forskolin biosynthesis. So this is generally it's under control studies, what is the load of this inoculum within the plant system, and these are the what we call it as biocontrol properties of this fungus. And also the major constraint is even the nematode attack for the roots actually. So even upon treatment with this fungus, so even most of the nematodes has been also been controlled within the field condition. So apart from that, next step is, can we have the compatibility of these endophytes with the Trichoderma viridae? So this is the 3 compatibility that we have noticed in that. So in that field studies which has been conducted with the Trichoderma viridae along with the 3 fungal system what we have isolated or identified. So and we have noticed again there is a good compatibility or a coherence that has been exhibited by these 3 fungus and without competing each other actually. And then in fact the combinations of T5, T6 and T7 which has really shown that is an impact on this even they work synergistically with even Trichoderma viridae also. So this is what its expression along with that same even acyl transferase has also been enhanced, and also in a CYP76 also cytochromes also been enhanced with that upon treatment along with the TPS 1, 2 and 3 and 4 actually. So with this then how do we take this into the application mode actually? So then we have developed a carrier based endophytic bioformulations for coleus forskolin cultivation. In that cases we have developed both the wheat bran based and also talc based formulations using this probiotic system, and then all of this has been grown and they are growing on a wheat bran as a very efficiently using it as a substratum, and then this has been applications, and we have noticed that one is even individually and also in combinations. There have been wheat bran based formulations and talc based formulations, and wheat bran based formulations are quite stable and also able to induce more amount of forskolin content than talc based formulations. And then shelf life of this one almost it's 6 months. It is highly viable of all this probiotic formulations. So among which like it is RF1 and then ESF1 are quite stable than ESF2. So with this just at the conclusion, Endophytes play a key role in enhancing the primary productivity as well as secondary metabolite production in the medicinal plants, and deployment of functional endophytes for cultivation of forcefully could be beneficial, and the concept could also be exploited for cultivation of other economically important medicinal plants. And successful compatibility was also noticed with the co-inoculation of fungal endophytes with Trichoderma viridae under field conditions. Besides rendering the plant growth traits, this approach could attributed to in-plant augmentation of forceful in biosynthesis in roots and reduced incidence of nematode infections in Coleus forcefully. Thus, the deployment of a specific microbial combination in present-day agriculture could be a value-added and a sustainable approach for the cultivation of economically important medicinal plants. This is we have carried out similar kind of an experiments with other medicinal plants like opium poppy, catharanthus, and also ashwagandha. And then there's a cross functionality of these endophytes also we have established with other commercial cultivations of aromatic crops, and also even Andrographis paniculata, so on Davana. So these are the publications which comes out on that, and anybody interested on this, please look into this and the details. And these cultures has been already deposited under National Culture of Industrial Microorganisms, and these are available for every researcher. Anybody can procure it actually. So with this I conclude, and then my sincere thanks to CSRCMAP and also the financial support from NWP 12th Fire Plant Project Network Project. And thank you, sir. Thank you for the audience, and also thank you for the organizers for providing me a platform to share my thoughts on this. So thank you. Any questions? I am open for the discussion, please.
Thank you, sir, for a good presentation. Now the topic is open for discussion. Okay, if you don't have any clarification, we will move on to the second oral presentation by Dr. Rajendran. He is going to deliver on effect of chitomium against tomato wilt complex incited by Fusarium axisporum, former species Lycopersicea, and Merodogaina incognita. Dr. Rajendran.
Sir, audible, sir?
Yes, clear.
So topic is unraveling the potential of bacterial endophytes and its practical implications, sir. Hello, sir, audible? Hello? Sir, audible?
Yes, it's audible, sir.
Yes, sir.
It's audible.
So topic is changed, sir. So I have given the unraveling the potential of bacterial endophytes and practical implications. Hello, shall I proceed, sir? Hello?
Hello.
Yes, you can proceed.
Okay, thank you, sir. Respected chairman and rapporteurs and scientists all over India. I'm sharing the work experience of our previous and current our projects. So initially around 2000 year, we have concentrated mostly on the rhizosphere, phyllosphere, and spermosphere organism for exploitation, and also mostly we are concentrating on the plant growth promoting rhizobacteria. Then followed by after 2005, we have concentrated on the plant growth promoting endophytic bacteria from cotton and coconut and other crops. Then due to their stability, and we have isolated from different stem, flower, and leaf and root region. Followed by now they are discussing scientist forum what they are telling that the seed, the microorganism present in the seed is highly viable, and it is a seed generation to generation transmission and vertical transmission high. That's why we are now concentrating on the bacterial endophytes from the seeds, mostly for the youngsters and student friends. When they are going for the isolation of microbes, mostly we are going for the culture dependent analysis, where we are getting the 10% of the culturable organism. Remaining 90-80%, we have to go for the independent the metagenomics. It is very costly. While doing the culture independent dependent analysis, mostly we are doing the isolation, identification by the morphological, biochemical, and molecular characterization using the 6' rRNA partial base pair, that is around 600 base pair sequences. And we are not concentrating mainly on the molecular characterization up to this whole genome sequencing. Nowadays, they are stressing the proper identity of the organism. So initially what we'll do, go for the 100 and 200, 300 isolates from the rhizosphere or endosphere, seed endophyte. We are concentrating on the antagonistic activity. Then we are testing as a growth promotion activity as a phytostimulation. Then followed by if it is doing good in the antagonistic activity, then we are going for the roll-towel method and bioformulation. At the end of the day, when you go for the molecular characterization, we are lacking there because we are already partially sequenced. So you have to give the importance for the proper identity of the antagonist at initial level and doing this split technique. That is two methods we are following for the endophytes: split technique and serial dilution technique, where we have to maintain the proper sterility check has to be maintained for the proper isolation, proper identity. Then when you give the get this type of organism around 20, 30, 40 enormous bacteria will be unique bacteria will be isolated, and morphologically you are going for the identification. Then see, don't rely on the single method of antagonistic activity, only dual culture technique. You have to go for the multi-number of triangle method, culture filtrate level of screening, and we can easily select the good candidate just to not rely on the single method of dual culture technique alone. This is our Bacillus subtilis isolated from the zerbera. This is endophytes from the groundnut, where we have given this dual culture triangle method, then culture filtrate assay, and some we can see the role. This is dual culture technique. When we see these two strain are giving the high percentage of control, that is EGN1 and EGN4. And then remaining strains are very less antagonistic activity. When you go for a triangle method, also you are giving the 100% inhibition, EGN1 and EGN4, and also it is comparable with the standard isolates. So once your isolate, that is a sclerosis alpsicaceae stem rot of groundnut, where it produces abundant of sclerosium. When you see that particular endophyte, it can arrest the total inhibition of the mycelium and also no production of the nil production of the sclerosia. Whereas within 16 days, you can see the 600, 300 sclerosal bodies in the petri plate itself. That's why selection of proper candidate is, even if it's failure in that dual culture, you can go for the paper filter paper disc assay when they see the modification of mycelium and thickening or thin growth of the mycelium, like that. 3 or 4 method you have to employ for the selection of the good candidate under in vitro, not rely on the single method. Here you can go for the culture filtrate and amending 2:1 ratio in the media, and you can assess the 100% control of the that sclerosis alpha. It is a virulent pathogen and causing stem rot. If it is giving only the inhibition is more, shall we go for the formulation? No, it has to be go for the phytostimulation activity. Earlier we rely only on the plant growth promotion by the roll-towel method. You have to simply employ this CEDRA4, phosphorus solubilization, proteolytic activity, solulase activity, ISA and IA activity under laboratory. Within 2 or 3 days, we can screen around 20 to 30 isolates or enormous isolate. And if you go for the roll-towel method, it will take minimum 14 days for your result. Within that time, you can go for the mass screening by using the CEDRA4 or ion chelating activity. You can see this eye activity in the Bacillus subtilis. Here is the Pseudomonas species. Finally, we conclude this Pseudomonas species. This is Acinetobacter activity. And then you can see the phosphate solubilizing activity. Most of our soils are fixed with phosphorus. To have this type of activity, that has to be screened under laboratory condition. When you see this two strain in vitro, it is giving good, that is antagonist activity is EGN4 is good. When you go for the phosphate solubilizing activity, EGN1 is giving high allozone. So selection of good candidate is important. Don't rely simply on the this biochemical test, where we can do the laboratory. It has to be confirmed. This for thesis or our project work is enough, but once we go for the field level trial, it has to be clearly mentioned with the supporting data. And also we have analyzed the growth promotion in the groundnut by roll towel method. You can see region 1, and that is the Pseudomonas species, and Acinetobacter, they are giving around 4,200 in Veger entice compared to the control. When you analyze this in the rhizoscanner architecture, whether it is giving good architecture, root architecture or not, yes, it is that Pseudomonas species are giving more 3,370 root length with more number of root tips compared to other Bacillus or Acinetobacter, where it indicates the more nutrients absorption, and it can survive in the rain-fed condition. That much data you have to generate for release of your new strains. And also thermostable compounds. To support our strain, we have to go for the thermostable compound identification, where we have to get the culture filtrate and amend with the media and go for boiling, sterilization. After that, you have to add and check under the in vitro, where it produces the high level of inhibition compared to other strain. Based on this, you can identify the good candidate under in vitro. Whereas in this our Bacillus subtilis strain, already we have sequenced that's lipopeptide antibiotic genes, and we have we got the NCBI accession number. And also for this BBV Bacillus subtilis strain, we have noticed the presence of uterine A, D, surfactant gene, bacillomycin, bacillicin, fungicin, and ericin. So many antibiotic, enormous antibiotic producer is the Bacillus subtilis, we have noticed. And also the presence of foreign sensing regulatory genes in the Bacillus subtilis for the easy cell-to-cell communication, and also the their survival in the endophyte, and also formation of biofilm. And also they have the excellent exopolysaccharide by based on the 15% STS page analysis we confirmed. And also it has more biofilm activity, on par with the Bacillus subtilis standard isolate. So this type of analysis, when you do the under laboratory condition, the data will be supported for your further registration. And also when you go for whether it is it has nematicidal activity against root knot nematode, we have checked for the Bacillus subtilis. When you treat the 25%, 50%, 100% concentration culture filtrate, when you increase the concentration, you can see the egg hatching capacity has been reduced, and the abnormal and darkened egg matrix has been seen in the SEM study. And also the most of the juveniles 2 stage has been died due to this culture filtrate assay. When you see the egg under a SEM study, you can see the malformed and shriveled eggs, and also the female treated female has been shrunken and misshapen female. So totally we recorded 87 dead juveniles in the undertreated nematodes. After that, when you go for the secondary metabolite production as a ethyl acetate fraction, it has the metabolites for all the EGN1, EGN4, and also Bacillus subtilis, and surfactant here, phenazine, phylonetin, everything we confirmed under TLC. When you go for the GCMS study for Bacillus subtilis and this APCO, that is Bacillus subtilis, we can see the peptidoglycanic acid, pyrrolol compounds, then butanoic acid. These are all the antibacterial and antifungal and nematicidal property has been recorded for these 3 elite strains. You can see the common that is acrylonitrile, then oleic acid, then pyrrolol compounds are present in the EGN1, EGN4. When you go for this molecular characterization, we are only rely on the partial sequencing. We are submitting and say Bacillus subtilis, Bacillus amelia. Once you go for the higher base pair, more the targeting more than 1000 base pair, when you give it is Bacillus cereus, means it is referred as causing some allergic reaction to human being. Pseudomonas aeruginosa means allergic reaction. At that point, we are struggling. That's why initially we have to go for the selecting good candidate with proper identity. Has to be strengthened. Otherwise, at the end, we are facing a lot of problem for the formulation, registration, and commercialization. That's why for any screening of this good antagonistic endophytes, you have to go for proper screening, then growth promotion, that and then antagonist activity with different methods, and combined all this data and identify with the more than 1 kb base pair. And also we have to see that endophytes is surveying on the sieve tube or not. Yes, it's confirmed under the SEM only after 10 days after treatment, seed treatment. It has to be periodically seen up to the seed to seed, whether it is surveying in the next generation seed. That study has to be strengthened. And this is results of pot culture, where it gives the good growth promotion and with less disease incidence. And we have also the field data, but to have the practical implications in the challenge faced by these endophytes and PGPRs under laboratory conditions, we have to properly identify the particular strain by the designing the scar marker specific to Pseudomonas fluorescens or Pseudomonas fluorapis or Bacillus subtilis or Bacillus amyloidic efficiency. And also we can't go for all the isolate for the whole genome sequencing at the end. So properly select single antagonist for the whole genome sequencing. Already it is in process for our BBVV57. It is having an MRCL activity and also in vitro for activity for the fusarium isolate. And finally, toxicology data is highly essential. These are all the costly and highly expensive. So when you do the laboratory experiment, we can publish in the NAS rating 6 or one impact factor journal. When you go for registration, commercialization, where we are facing the proper identity. Hence, I acknowledge the biocontrol lab, first lab of Department of TNE for perform this research, and also I, as a team, I have to give the compliments for all my director, Dean, DSW, students, friends, and laboratory, everything. And also I thank our chairman and rapporteurs for giving me this opportunity to present our work. Thank you, sir.
Thank you, sir, for good presentation. Now the topic is open for discussion. If any clarification?
So I have one clarification, sir.
Yes.
Hello. So thank you for your nice presentation. It is really informative. But the doubt is that whether you have tested against some non-target organism, say beneficial organisms. So harmful organism, okay. So your biocontrol agent.
Yes, sir.
Okay, whether you have tested its efficacy or if it's effect on non-target organism or say beneficial organism.
Against Pseudomonas fluorescens or against Bacillus, like that you are asking, sir?
No. Say a non-target organism, I'm talking about, say, some parasite or say predators or say natural enemies.
Natural enemies, we have reported that the part and secondary metabolites or the jasmonic acid pathway, they will attract the natural enemies, sir.
Okay.
In our case.
Yes, that's why actually
after treating,
yes,
treating with the seeds and that attracts the natural enemies.
But that means these are safe to the natural enemies. Your biocontrol agent.
Yes, safe to natural enemies. But once you go for registration, there is a problem exists. Even though we are claiming all this data, they rely only on the NCBI whole genome sequence. Now the era is they are they want the entire whole genome sequence for our particular bioagents. Okay. They won't accept the mere 600 base pair or 700 base pair.
Oh, that's why. Yes. Thank you.
Thank you.
Okay.
I request chairman and co-chairman to summarize the session.
Okay. Finally, I thank all the speakers for excellent presentation with valuable. For one of the lead speaker by Rashmi Agarwal presented is mainly focused on holistic view of the molecules for further studies, and also mainly concentrated on genomics, proteomics, and also metabolics are the three core omics technologies, and also focused on bioinformatic tools for the use of potential genes. Madam also focused on the review of the genomic sequence of the biocontrol agents. Then another interesting topic by Dr. S. Nakiran, that is one of the most very good presentation in this session. That is one of the important topic, that is a bacterial endophytes. Okay, it is a need of an hour. I think the organizer have selected a very good topic for the presentation. So I compliment the Dr. Nakiran Saab for the nice presentation with the management of the bacterial and fungal and viral pathogens by using endophytes. But he also collected the bacterial endophytes against the pathogens. So I compliment him for the isolation of the 10 isolates, that is for the inhibition of the Phytophthora infestans. And also he concentrated more on hyperparasitic nature of the bacillus species on Phytophthora infestans, especially for the lysis of the sporangia and mycelium. That is one of the great contribution. Thank you, sir, for excellent presentations. And also thanks to the other two speakers, Vivek Babu and Rajendran, for excellent presentation with regard to the plant probiotic fungal endophytes and also the Chaetomium. And finally, I thanks to the organizer for giving an opportunity to chair the sessions. And also thanks co-chairman for valuable support for completion of this session. Similarly, I thanks to the rapporteurs, Dr. Sudarshan, Dr. Karim, for supporting the session and successful completion of the presentations. Thank you, sir. I hand over to the co-chairman for his remarks.
Okay, good evening to all. So first of all, I'm really thankful to the organizing committee of this National Symposium of ISMPP for the opportunity to act as a co-chairman of this technical session on New Horizon in Biological Management of Plant Diseases. So I sincerely thank the respected chairman, Dr. Thamaiya sir, the Dean College of Horticulture, Aravali, for meticulously handling the session. And also thanks to the rapporteur, Dr. Sudarshan from Mysore, and Dr. Abdul Karim of Sirsi. So here what happened, the lead lecture delivered by two eminent scientists of the country, Dr. Rashmi Madam and Dr. Nakiran sir, these were very informative and really knowledgeable. The work actually reflect the hard and dedication toward the science of New Horizon in Biological Management of Plant Diseases. Both the presentation paves the way for many new researchable issues. And in the coming days, I may request your kind support in this regard. So definitely, I'm sure they will definitely help us. So this was followed by two presentations delivered by Dr. Babu on probiotic endophytes and another presentation by Dr. Rajendran. Both were very informative and really these are praiseworthy. So I'm happy to see Dr. K.K. Bishwas sir, who is now also an adjunct professor of my university. Sir, throughout the session, we're very active. So I'm really happy to see you. And thank you once again to the organizing committee, chairman, and the participant. Thank you.
With that, we have come to the end of today's session. On the behalf of the organizing committee, I would like to thank the chairman and co-chairman and the rapporteurs for leading the session. I would also like to thank the speakers who have shared their important research findings with us in this platform.
Sir, with this, we conclude today's session. Tomorrow we have two technical sessions. The first technical session is on fungicides and the second is on ecology and epidemiology. First session starts at 9:30 and the second session will close on 3 o'clock, and from 3 to 4 we have the valedictory function. Okay, with this we conclude today's session. Thank you all.
Thank you.
Thank you.
Who will give the conclusion remark?
Yes, sir. Vishwas, sir.
Conclusion remarks will be given by the chairman and co-chairman.
It has been given, sir.
Hey, Dr. Kajal, they have given.
Given, sir.
No, okay. Nice presentation. I am very much appreciate and enjoy. Okay. Thank you very much to all.
Yes, sir. Everything is done for today. We shall meet tomorrow, sir.
Okay.
Thank you.
Thank you.